Hello everyone, Namaskar. Warm welcome to one and all. This is part 5 of chapter 6. The content to be discussed in this part, that will be, we have to differentiate monocot stem and dicot stem. The detail of monocot stem and dicot stem already we have discussed in part 5. This is, and then we have to see, that is dicot and monocot leaf, the structure, detailed structure, and then we have to differentiate, that is dicot and monocot leaf. This is part 5, chapter 6, anatomy of flowering plant. In part 4, we have discussed anatomy of dicot and monocot stem, and then that is dicot root and monocot root. Already we have differentiated dicot and monocot root. We have to see differences between dicot stem and monocot stem. You see, in a tabulated form I had given all important characteristics, but even then I like to repeat, that is what we have done in part 4, a part of this. Dicot stem, monocot stem. Children, stem the most important one, that is one important point, that there is, the hairs are there, hairs if are present, the hairs are always, these are multicellular one, that the vascular bundle, very important point, vascular bundle both in dicot and monocot stem, vascular bundles are, that is conjoint, and the xylem is enlarged. You have to see these are the basic points, but remember either this is dicot stem or monocot stem. So I can make it here that this is dicot stem here, that this is monocot stem. What is here? That is the multicellular hairs are present here, here the hairs are absent. Here we have that this is hypodermis. Hypodermis in this case, this one is, can you remember? That one is, yes, this one is 𝕆𝕋𝔼𝔼ℍ𝔼. Yes, hypodermis is there. Is it right? Well, in this case, the hypodermis is present below the epidermis. This hypodermis is, that is 𝕆𝕋𝔼𝔼ℍ𝔼 hypodermis. Now here we have, this is the endodermis, and here we have the vascular bundles arranged in a ring. If you are remembering, here we do not have, that is this one is, what is this? This one is the cortex, this is the cortex, and here then we have endodermis, then we have that is pericycle, and then at the center we have that is pith. You must be remembering all that, right? While in this case, that is ground tissue system is undifferentiated, and we have so many vascular bundles found scattered, not arranged in a ring. Here the vascular bundles are arranged in a ring, and the important here is, that is in between the primary phloem and primary xylem, the cambium is present, that's why the vascular bundles are open. Just a rough sketch I had given to you just to recall you, but we can differentiate it now. Dicot stem, come on, this is monocot stem. The first one is epidermis, it is single layered with trichome, that is trichome, these are, that is multicellular, that is hairs are present. Is it right? But usually that's why the epidermis, this one is rough, while in this case the single layered, it is without trichome, and usually the epidermis, this one is, that is smooth one. You have seen that the stem of maize or stem of, that is sugarcane, they have smooth, that is surface, because the trichome is absent here. Come to the next now, hypodermis. In this also I told you, just below the epidermis this one is, and the hypodermis this one is 𝕆𝕋𝔼𝔼ℍ𝔼. Here the hypodermis, that one is 𝕆𝕋𝔼𝔼ℍ𝔼. Cortex made up of several layers, parenchymatous cells, endodermis is there. What we mean by that is below hypodermis or even hypodermis, that is here the ground tissue system is differentiated. It is having hypodermis, it is having, that is cortex, it is having endodermis and pericycle, while in this case it is absent, cortex is absent, parenchymatous ground tissue is present from hypodermis to the center of the stem, and all this is, so we can write ground tissue system, we can write that one is, yes, that is undifferentiated. Is it right? Okay. Come to the next now, that is pericycle. Here it is made up of parenchymatous and 𝕆𝕋𝔼𝔼ℍ𝔼 cell. Definitely pericycle is absent. Vascular bundle, the most important part here is about vascular bundle. Almost all are of uniform size, larger towards center, smaller towards periphery. You know that these are arranged in a ring, these are scattered, these are conjoint and open. This is not the difference. These are conjoint, but these are closed. Closed we mean by that is cambium is absent. Cambium is absent. Is it right? Cambium is absent. Bundle sheath is absent. Here vascular bundle is surrounded by 𝕆𝕋𝔼𝔼 bundle sheath. You know, this is that is vascular bundle, and here we have, this is the bundle sheath we have here. This is the xylem, vessels arranged in V shape, and this one is the phloem. This is the phloem, is it? And this one is xylem. Come to the next now. That is phloem parenchyma is present in phloem. Usually it is absent. Water containing cavity is absent. Here water containing cavity is present. Here if you are large here, that is xylem vessels. These are xylem vessels. These are like this. This is protoxylem, and close to protoxylem, this water cavity is present. Water cavity. Is it right? Here well-developed pith is present here at the center. It is absent in this case. Medullary range definitely. Here in between the vascular bundles, that these are the vascular bundles we have here. Here in between the vascular bundle, this is endodermis. This one is pith. So here these are the medullary rays are present in between the vascular bundle. It is absent. The vascular bundle, this is known as conjoint, collateral and open. Therefore it is known as 𝕍𝕀𝔻𝔼𝕆, conjoint, collateral and closed. This is called as atectostele. Is it? One more difference you can see here, that is xylem vessel. Here in this case, the xylem vessels, these are usually polygonal. These are like this, and these are linearly arranged. While here, the xylem vessels, these are round. That is xylem vessels, and these vessels are arranged either in V shape or in Y shape. Is it right? Visualize it. Now the third part, anatomy of that is leaves, dicot leaf, and that we have to go for monocot leaf. You know dicot leaf, morphology I told you, we call it now. The leaf is known as dorsiventral leaf. Dorsiventral leaf, what we mean by? You see here in case of dicot leaf, if this is the dicot leaf, children, you know dicot leaf is like this. It is having this, the midvein is there, and this that is reticulate venation is there. One surface is upper surface, the other surface is lower surface. Therefore, the leaf is known as dorsiventral one, upper surface, and this one is that is lower surface, right? Upper surface in case of dicot, it is known as that is abaxial is the lower one, and upper surface is known as adaxial. So this surface is adaxial one. This surface is adaxial one. So leaf is called dorsiventral. While in case of monocot, you know, that is monocot, the leaves are like this. Leaves are arranged like this. That is, they do not have upper and lower surface. Almost both the surfaces are equally, that is good. This leaf is known as bilateral leaf in case of that is monocot. This is morphology. Morphology is very well related to the internal structure of this. Okay, come to this now. That is anatomy of that is dorsoventral leaf. See the diagram here. This is the leaf. That is leaf is this is the leaf here. It is packed in between the two layer upper and the lower epidermis. This is the upper epidermis. This one is the lower epidermis. In between that is the leaf's tissue is present. Okay. So upper epidermis and lower epidermis. The vertical section we have to cut. Dorsoventral leaf through lamina. It shows that is three main parts. That is upper epidermis, lower epidermis, and the tissue in between this, that is known as mesophyll, and in mesophyll we have that is vascular tissue. So the three important parts we have here, that is epidermis, mesophyll and vascular tissue system. Epidermis, it covers both upper and lower surface. This is upper surface, this one is lower surface. Upper epidermis is known as I told you, it is called what adaxial, it is called adaxial epidermis, and this is called that is abaxial epidermis. See here, lower epidermis is abaxial, and that is both surfaces they have cuticle to reduce that is rate of transpiration. Abaxial surface in case of that is dorsal. Dorsal, that is the dorsoventral leaf is like this. It is having a bulging here which represents the that is mid-vein. On lower side on abaxial surface, that is it bears more number of stomata. Stomata here are less, here the number of stomata are more, that is than adaxial surface. Okay. On that is upper side, either the epidermis that is stomata are less or even absent. Mesophyll, as I told you, in between the two epidermis, the tissue present between the upper and lower epidermis is called mesophyll. Mesophyll possesses chloroplast. It is made up of parenchymatous cells, and that will do photosynthesis. It is made up of parenchyma. This mesophyll, this is made up of these parenchymatous cells, and these have the chloroplast inside, which will do photosynthesis. In case of monocot, in case of that is dicot leaf, this mesophyll that is differentiated into that is upper part and the lower part. The cells of the upper part are columnar like this, parenchymatous cell, columnar like this without intercellular space, and these are perpendicular to the that is surface. This tissue is called palisade parenchyma. This is towards upper side. They have more number of the chloroplasts because the light will fall on the upper surface of the leaf through photosynthesis, and the lower part of the mesophyll, that is in between palisade and the lower epidermis, we have the parenchyma, that is spongy tissue, which have more intercellular space, that is here spongy tissue. Adaxial placed palisade parenchyma, that is the same thing what I told you right now, that is elongated cells which are arranged vertically parallel to each other, and these cells are oval, round or loosely arranged. Spongy parenchyma we have here below the palisade tissue. Intercellular space that will help in gaseous exchange. Large number of spaces are there. Air cavities are there between these cells. It is also called as parenchyma. Okay. You can see a beautiful diagram we have here. That is in case of dicot leaf common. That is here. This is the adaxial surface. That is this one is common. This one is that is this upper surface. This is adaxial. It is having the cuticle over it. This is the cuticle. It is important. On all aerial part the cuticle is there. Then this is the epidermis one. Stomata are usually absent on upper side, and this is the this whole tissue is known as this is mesophyll. As I told you that a mesophyll, it is having the upper part. This one is what is this? This one is the palisade mesophyll. Cells are elongated, having more chloroplast perpendicular to the surface, and they will carry out more photosynthesis. In between palisade and that is this is the lower epidermis. This is the parenchyma we have. Sufficient air space is there, known as air cavity, also known as that is the spongy mesophyll. On lower epidermis, that is abaxial surface, more stomata are present on abaxial surface to do that is gaseous exchange. Is it? One part is cuticle, the second part is that is mesophyll, divisible into that is palisade and that is spongy tissue. Come to the vascular tissue, very important one. Vascular tissue, see, in case of dicot leaf, you see in vertical section, we will see a bulging is there. This section, the leaf we have here, this is the mid-vein. If the section will be cut through this, then an area will come here which will represent the mid-vein. So here the bulging is found. It represents the area where the mid-vein is present. No doubt that the other vascular tissue will also be there, which will not be of the same thickness. The vascular bundle which is found, which represents the mid-vein, is the thickest one. So here sort of bulging is there. Important thing, vascular bundles can be seen in veins and midrib. This is the midrib, these are the veins. Size of the vascular bundle depends on the size of the veins. The vein vary in thickness, as I told you right now, in reticulate venation. The vascular tissue vascular bundle in the mid-vein region is the thickest one. The vascular bundle in the mid-vein is the largest, thickest one. Vascular bundle, very important. Vascular bundle are consonant like stem, is it? And these are closed. Remember, either this is dicot leaf or this is monocot leaf. Remember, cambium is always absent. Otherwise, if the cambium is there, it may give secondary growth and the leaf will increase in thickness. You will see that leaf will increase in thickness. No, never. That's why vascular bundle are consonant and these are that is closed. We can say vascular bundle, these are almost similar to that is monocot stem, because in monocot stem also the vascular bundles are consonant and closed. Okay, before we proceed to this monocot leaf, we have to see detail of this dicot. One more thing which is very interesting one. You can see here this is vascular bundle. Vascular bundle, this one is. What are you seeing here, vascular bundle? That is one very important point. That you see that this one is where we have xylem. Xylem is here. Where we have this is xylem. Xylem is here and this one is the phloem. This is very important point. What you see here, very, very important point. This one is this vascular bundle. These are surrounded by parenchymatous bundle sheath. And very important point, this one is each vascular bundle consists of xylem, which is towards adaxial side, and the phloem is towards lower side. You see here, you can remember here that this is vascular bundle. So which side is abaxial one? This side is abaxial one. This is abaxial side, and this side is that is adaxial side. Don't do this mistake. This is very important in examination. So xylem lies towards adaxial epidermis. Here is the xylem. Xylem will be like this. That is, this is metaxylem. This is protoxylem. And the phloem that lies towards that is lower epidermis towards that is abaxial side. Remember, children, this one is phloem, is it? And this one is that is xylem. So vascular bundle, because these are jointed together, this is conjoint. Cambium is absent, that's why this is closed. And one more time I am telling you here, phloem is towards lower side, abaxial side, and the xylem is towards adaxial side. This will be the protoxylem. The protoxylem, it faces towards that is upper epidermis. Is it clear? Come to the anatomy of monocot leaf. Is it? Monocot leaf. You see now, before we proceed, see monocot leaf. This monocot leaf, you know that sometimes it may have one that is thick vein. It is having so many that is veins of the same thickness. That is, there is parallel venation is there. You see, parallel venation, that is related to that is anatomy of that is monocot leaf. As usual, this is adaxial epidermis. This is abaxial epidermis. Number of stomata that is are almost equal on both sides. In case of dicot, more stomata were on abaxial side. Here, that is number of stomata are almost equal on both sides. Here and here, that is stomata. Is it right? This is upper epidermis, lower epidermis. One important point we have here, that is in some grasses on upper epidermis, some of the epidermal cells, they will become enlarged like this. And these cells are known as bulliform cells. What are these? These are called bulliform cells. These are enlarged one. We have to see the function of this. Now this is the mesophyll. You know, mesophyll in between upper and the lower epidermis. Does it has that is the palisade and sponge? No. This is the difference. Mesophyll doesn't have that is palisade. This is undifferentiated one. Mesophyll is that is undifferentiated. Will you remember? Important. Then here all these cells they have that is chloroplast will do that is photosynthesis. This is one of the vascular bundle we have enlarged. What is this? This is the xylem. Xylem is towards upper epidermis. Protoxylem is towards upper epidermis. This one is metaxylem. Can you remember? This one is metaxylem. This one is protoxylem. This one is, and here this one is phloem. So vascular bundle, because phloem and xylem, these are jointed together. Vascular bundle will be constant, and this is closed because the canyon is absent. Can you summarize it now? Come on. Anatomy of monocot leaf. I told you the leaf is isobilateral leaf. Is it? Isobilateral leaf anatomy is almost similar to that of dorsiventral leaf, but the differences are there. Some characteristic differences. Can you summarize? Isobilateral leaf. Yes, I told you, remember, stomata are present on both sides, almost equal number of stomata are on both sides. In grasses, very important point now, certain adaxial epidermal cells that they modified themselves into large empty colorless cell, and these are called what? Bulliform cells. What are these? Bulliform. Very important point, bulliform cells. That is, bulliform cells, what the function? When bulliform cells of the leaves, they have absorbed water, they will be full of water. They will be full of water. There is no scarcity of water. These are turgid. When these are full of water, these are turgid, and the leaf surface that will be exposed like this. That will open. The leaf will be open. When there will be scarcity of water, then these cells will become flaccid, because already scarcity of water is there. That's why they will lose the water. They will become flaccid, and in that case, leaf will be rolled up just to reduce transpiration. See it now. The leaf surface is exposed when they are flaccid, when the scarcity of water is there, water stress is there. They make the leaves curled inward like this, so that the stomata will be inside and the water loss will be minimized. Next point, important difference. Mesophyll is not differentiated into palisade and spongy, because there is no need. In this case, that is, the leaf will get light from all the sides, almost equal intensity of the light will be there. That's why there is no need to concentrate photosynthetic tissue on one side, just like dicot leaf. That is not differentiated. Parallel venation is there. This is reflected near to similar size of the vascular bundles are there. That is definitely, that is main vein is there, that may be little thicker. Otherwise, all the other veins that will be of similar size. Right? Yes. So, what we have seen by now, that we have to summarize it in the form of table form, differences in anatomy of the dicot and the monocot leaf, characters, dicot and monocot leaf, that is in a fast way we can go. Type of the leaf, dorsiventral, yes, no need to go like this. This is dorsiventral one mid vein is there, reticulate venation is there. Isobilateral leaf, this is, I told you detail, stomata, usually more on lower side, that is hypostomatic, equal stomata on both upper and lower side. Amphistomatic. Mesophyll is differentiated into palisade and spongy parenchyma. It is not differentiated. There is no need. Bulliform cells are absent. In this case, these are present, especially in some grasses. And the vascular bundles, due to presence of reticulate venation, vascular bundles are of different size. The mid vein will be, that is the thickest one. The side vein will be little, that is less thicker, and that will be the least thicker. While in this case of nearly similar size due to the presence of parallel venation. This is about anatomy, comparison of anatomy of dicot and monocot leaf. Then that way the part 5 is over. The part 6 we have to do, and in that part in the end we have to discuss, that is NCERT exemplar, and then the previous year examination question, because that will be the last part of chapter anatomy of flowering plant. Thank you.